Hey, hello everyone, this is Vishal Boyer. Today we will learn about peptides and proteins. So, how peptides are formed? Peptides are formed of the amino acid chains. So, let us see how these peptides are formed from the individual amino acid. As you can see in front of you, two amino acids, two different amino acid, like they are labeled as R1, that is the first amino acid, and the second R2. And over here, the very important reaction happens. Condensation reaction happens and a water molecule is removed, and you can see there is a bond formed between amino acid first, the carbonyl group of first amino acid, and the amino group of the second amino acid. So what we see over here, the bond which is formed is called as peptide bond, which is highlighted in yellow color. So this bond is very important, which forms in between two amino acid coming together, and in this way several amino acid can combine with each other with this peptide bond, and we call it peptide linkage. And with the peptide linkage we get amino acid bound, amino acid binding to each other. Like one example over here is pentapeptide seryl glycosyl tyrosyl alanine leucine. Now in this particular peptide we are observing five different amino acids coming together and forming a peptide bond with each other, and as a result we are getting a peptide. And in this particular way several amino acid can come together to form polypeptide. Over here, one end of a peptide or polypeptide is amino terminal because amino group is flanking over there, and the other end is called as carboxy terminal. And remember this thing very well, in any peptide or polypeptide there are two ends, one will be carboxyl end and one will be the amino end. Then we will see how the proteins which are formed from polypeptide chains, they are having different levels of protein structure. If you observe this diagram carefully, the very first structure, that is called a primary structure, is the simplest one, in which we are just observing the amino acid residues bound together with a peptide linkage. So it's just a simple chain. Then coming to the second structure, that is called a secondary structure in the protein organization, that is alpha helix or even beta pleated sheath is coming in the secondary structure. Now this alpha helices or beta pleated sheets can come together to form the third, that is tertiary structure, the well advanced structure in the protein. That is this particular tertiary structure. Now this is one of the assembly. Now this particular tertiary structure can come together and form an assembly that is called as quaternary structure, which is on the right hand side of your display. Now in this case hemoglobin is actually a quaternary structure, so there are many tertiary structure polypeptide chains are coming together to form the hemoglobin. So in this way we have learned about the peptides and polypeptides and how the proteins are having different structure. Thank you very much.